anatomical structures of the same. Requirements: young sunflower stem, maize stem, young fenugreek root, maize root, watch glasses, slides, blades, cover slips, brush, section lifter or forceps, safranin stain, blotting paper, and a compound microscope. Procedure: in order to study the transverse section of a root and stem of dicotyledonous plant and a transverse section of root and stem of monocotyledonous plant, you will have to prepare their slides. The preparation of a slide is carried out in the following three main steps: sectioning, staining, mounting the stain section. The first step is sectioning. For exposing the various types of tissue, you have to take transverse sections of the plant material. The transverse section is taken by passing the razor's edge or blade's edge at right angle to the longitudinal axis of the plant material. Let us see how we take the transverse sections. For taking transverse section, hold the plant material vertically between the thumb and the index finger of your left hand as shown here. Now, keep the edge of the razor or blade at right angles to the longitudinal axis of the plant material and cut thin sections. Transfer these sections from the edge of the blade with the help of a wet brush into a watch glass containing water. The next part of the preparation is staining. We use safranin for staining plant material. By staining the material in appropriate manner, we are able to distinguish different types of tissues from each other. Safranin stain specifically stains the lignified tissues. From the stain bottle containing safranin, take a few drops of stain in a watch glass. Put some water in the watch glass in order to dilute the stain. Dilute the stain with approximately equal number of water droplets. Select some 3 or 4 good, thin and transverse sections. Transfer these selected sections from the watch glass containing water to another watch glass containing safranin stain. Allow the sections to remain in the stain for about 2 to 3 minutes. After staining, wash the sections with water in another watch glass. Do this repeatedly for removing the extra stain, if any. Now comes the third step of mounting the stain section. For mounting, take a clean slide. Make sure that it is thoroughly clean. Place a drop of glycerine or water in the center of the slide. Glycerine acts as a good mounting medium and keeps the section moist. With the help of a brush, transfer a well-stained transverse section to this drop. Add some more mounting medium so that the stain does not dry up. Then place one side of the cover slip in such a way that it would touch the mounting medium on the slide. Support the cover slip with the help of a needle. Now lower the cover slip gradually and remove needle when it comes in contact with the mounting medium. After mounting the section, blot the excess of glycerine or water from the sides of the cover slip by using a clean blotting paper. While mounting, care should be taken not to allow air bubbles to enter the mounting medium. If you find the air bubble trapped inside the cover slip, repeat the procedure of placing the cover slip. Presence of air bubble in the mounting will obliterate the view of the mounting and thus take care to remove every air bubble if it gets trapped inside the mounting. Observations. We shall see transverse sections of sunflower stem, fenugreek root, maize stem and maize root. Sunflower and fenugreek are dicot plants while maize is a monocot. Therefore, we will find different types of anatomical structures in each of these plant parts. We pick up specimens of young sunflower and fenugreek so that they do not show secondary growth in their anatomical structure. Transverse section of sunflower stem. Place the prepared slide on stage of the compound microscope and now adjust the view of transverse section of sunflower stem. First of all, see the section under the low power of a compound microscope. Observe the following structures from outside inward. We can observe epidermis, hypodermis, cortex, endodermis, pericycle, stele, and pith as we see from outside to inward. The first layer that we can observe is epidermis. It is the outermost layer made up of brick-shaped cells. These cells are covered over by a thin cuticle. The cuticle is covered over by a number of multicellular structures called trichomes. The epidermis layer shows few stomata. The layer is interrupted when stomata is present. The function of epidermis is to protect the underlying internal tissues. The second layer is hypodermis. It is immediately below the epidermis. The hypodermis layer consists of 3 to 4 layers of collenchymatous cells. The collenchyma cells are spherical or round. The corners of the collenchymatous cells are thickened due to the deposition of cellulose and pectin. The main function of the collenchyma is to give mechanical strength to the organ in which it occurs. The region below hypodermis is called the cortex. Cortex is made up of 3 or more layers of parenchymatous cells and a few resin ducts. Each resin duct is surrounded by a layer of small secretory cells. The parenchyma consists of oval or spherical cells with intercellular spaces. The main function of parenchyma is to store food, water, and other substances. The next layer is called the endodermis. Endodermis is the innermost layer of the cortex. It consists of a single layer of cylindrical or barrel-shaped parenchymatous cells containing starch grains. The main function of endodermis is to regulate the passage of materials both into and out of the vascular system. Pericycle is the next structure which you can observe just internal to endodermis. Pericycle is a multi-layered structure. On the inner side of pericycle, there are vascular bundles. Pericycle is made up of 2 types of tissues. Just above the vascular bundles, it is made up of sclerenchyma, and in between, it is made up of parenchyma. The sclerenchymatous patches of pericycle, which lie just above the vascular bundles, are known as hard basts. Now let us observe stelae. The central core of tissue consisting of the vascular bundles is called stelae. In sunflower, which is a dicot plant, the vascular bundles are arranged in a ring in the stem. The vascular bundles of sunflower stem are conjoint, collateral, and open. Let us understand the meanings of these words. Conjoint. When xylem and phloem are on the same radius, then the vascular bundle is called conjoint vascular bundle. Collateral. Xylem and phloem are present on the same radius in such a way that the xylem lies inwards and the phloem lies outwards. Such vascular bundle is called collateral vascular bundle. Open. In an open type of vascular bundle, cambium is present between the xylem and phloem. Such stems with open vascular bundle can show secondary growth due to this cambium. Each vascular bundle consists of phloem towards the periphery and xylem towards the center. The metaxylem lies towards the periphery, while protoxylem lies towards the center. Thus, xylem is endarc. The endarc xylem is a characteristic of a stem. The xylem and phloem of each vascular bundle is separated by a few layers of meristematic cells called cambium. Xylem and phloem are complex tissues because they consist of more than one type of cells. Xylem is made up of xylem vessels, tracheids, xylem parenchyma, and xylem fibers. The xylem transports water and mineral nutrients in plants. Phloem is made up of sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Phloem transports prepared food materials to various parts of the plant body. Each vascula is covered by few layers of sclerenchymatous cells forming a hard bast at its outer surface. Sclerenchyma is a dead tissue. The cell wall is lignified due to the deposition of lignin. It gives mechanical rigidity to the stem. The innermost last part is the pith. It is enclosed by the vascular bundles. There is a wide pith made up of parenchyma. Transverse section of young fenugreek dicot root. Just as in case of sunflower stem, take the sections of young fenugreek root. Stain and keep the slide ready for observation. You will have to observe this slide of transverse section of young fenugreek root under the low power of a compound microscope. Start observing the anatomical structures from the outside inward. In this case too, you shall observe epidermis, cortex, endodermis, pericycle, stelae, pith, and conjunctive tissue as you observe from outside to inward. Epidermis is also called epiblemma. This is the outermost layer made up of a single layer of thin-walled tubular cells. Some of the epidermal cells bear root hairs. They are unicellular hair-like structures. With the help of these root hairs, the root absorbs water and mineral salts from the soil. The second part is cortex. Cortex lies just below the epiblemma. It is made up of many layers of parenchyma containing starch grains. The third layer is endodermis. The cortex is delimited by this single-layered endodermis. Endodermis is made up of barrel cells. The radial walls of the endodermal cells show thickenings of lignin and suberin in the form of dots. These dots are called Casparian dots. Next is the pericycle. Pericycle is made up of a single layer of thin-walled cells. It lies just internal to the endodermis. Stelae of root consists of radially arranged 4 xylem strands alternating with 4 phloem strands. Such type of stele is called tetrarch. In each xylem strand, the metaxylem lies towards the center and the protoxylem towards the periphery. Thus, the xylem is also described as exarch. Pith is the small region in the center which is made up of parenchymatous cells. Pith is enclosed by the xylem and phloem strands. We can also observe conjunctive tissue. This tissue lies in between and around the xylem and phloem. The conjunctive tissue is made up of parenchyma. Transverse section of maize stem. Let us now understand the anatomical structure of monocot stem and monocot root. Maize is a monocot. Let us observe this transverse section of maize stem under the low power of a compound microscope. While watching the slide from outside to inward, we come across following regions: epidermis, hypodermis, ground tissue, vascular bundles. Epidermis is the outermost layer made up of a single layer of cells with thick cuticle and stomata. The cells of the epidermis lack hair-like outgrowths. The second region, the hypodermis, lies just below the epidermis and is made up of a few layers of sclerenchyma forming a narrow zone. Inside this, there is a ground tissue. Ground tissue means the entire tissue except the vascular bundles that are enclosed by the hypodermis. Ground tissue is made up of parenchyma. Vascular bundles of maize stem are seen running through the parenchymatous ground tissue. Many vascular bundles which lie scattered in the ground tissue. Each vascular bundle is conjoint, collateral, and closed and is bounded by a sclerenchymatous bundle sheath. Closed vascular bundle is the one that is devoid of cambium. Thus, in monocot stem, there is no secondary growth. The xylem is Y-shaped. The arms of Y are occupied by large metaxylem vessels, while the stem is made up of two small protoxylem vessels. In between the vessels, there are tracheids. The innermost protoxylem disintegrates and forms a cavity which is known as lysogenous cavity or lacuna. The phloem, which lies just above the xylem, consists of sieve tubes and companion cells. Transverse section of maize root. The last specimen which we are going to study is monocotyledonous root of maize. Let us observe the transverse section of maize root under the low power of a compound microscope. We can see the following structures as we observe from the outside to inward. These are epidermis, cortex, endodermis, vascular bundles, pericycle, conjunctive tissue, and pith. Epidermis, the outermost first layer, is made up of a single layer of thin-walled cells. Some of the cells of the epidermis grow out into root hairs. Next is the cortex. The young root of monocot shows the cortex made up of many layers of parenchymatous cells. But in older roots, a few layers of cortex lying below the epidermis become thick-walled and form a distinct zone called exodermis. Endodermis is the innermost layer of the cortex. The cells of the endodermis are thick-walled, but few cells called the passage cells, which lie just opposite the protoxylem, are not thick-walled. The inner and radial walls of the endodermal cells are more thickened than those of the outer walls. Vascular bundles of a monocot root are enclosed by the pericycle. We can see number of xylem and phloem strands which occur separately on different radii and alternate with each other. Thus, the vascular bundles of a monocot root are said to be radial. The xylem is exarch. As there are many xylem strands alternating with the phloem strands, the vascular bundles are called polyarch. Exarch. The xylem is exarch. Pericycle lies just internal to the endodermis and is made up of thin-walled cells. You can also see the conjunctive tissue. The tissue which lies in between and around the vascular bundles is called conjunctive tissue. The conjunctive tissue is made up of parenchyma, but in a mature root, the same tissue is lignified. Pith is enclosed by the vascular bundles. There is a large central pith made up of parenchyma.